Good afternoon. Thank you for coming. So I'll talk about some exciting technologies that we have to drive genomics forward. And it's all based on DNBSEQ technology. So first, Complete Genomics was founded in 2005. We just celebrated the 18th birthday in Silicon Valley, and with a goal to do affordable routine whole genome sequencing. We acquired 2013, and since then, and before that, over $1 billion is invested in this technology that we call DNBSEQ today. And the technology is used really globally. In Complete Genomics, in the US, we have about 200 scientists and lots of patents, almost 600. Lots of users, 6,000 papers, and lots of petabytes of sequence on this technology already. So I mentioned 6,000 publications in all different journals, from the top journals, covering all applications. Really, this technology is proven like any other NGS, MPS. We prefer massively parallel sequencing name. And there is a big growth, so it will continue. So one thing that we've realized and are providing, in addition to sequencing, we provide the complete solution for the complete workflow from sample preparation, library preparation, then obviously sequencing, and at the end, all this processing of data to get to VCF files. So this is important for easy adoption, for scaling, and for making sure that every part is affordable. Because we don't want $100 genome and $200 library prep. So that's our goal, to enable you to use everything cost effectively. So in terms of the sequencing, we really cover from the portable sequencer to the mainframe. Two sequences that I want to highlight in this talk. One is G99. It's the fastest massively parallel sequencing. You get paren 150 in 12 hours. Actually, you can get first reports in a couple of hours, for the infectious disease studies. And T7 is our most popular sequencer that can produce up to 60 genomes per day. But it has 4 flow cells. I think this is the only sequencer that has 4 flow cells, and they are completely independently running one from another. So it's practically having 4 sequencers together next to each other. So you can run in one flow cell 15 genomes, and obviously you can run similar or larger number of single cell where you need lots of reads. So it's very, very flexible and affordable. So this DNBSEQ technology runs on all our instruments. We don't have any deviation from that. Library prep is the same as any other approach. What we do at the end, we form circles and then run a rolling circle replication to generate about 200 copies as a concatenator. And we figure out conditions how this concatenator sort of folds in a really small nanoball, about 200 nanometers. So you can do all of these DNA nanoballs prepped in a single tube, billions of them. They repel each other. And then we just pipette them on a silicon chip with the sticky spots. So only one DNA nanoball sticks per spot. And more than one cannot fit there, so there is no overloading, over 95% occupancy with a single DNA nanoball. So it's a very, very efficient process, but it has many other advantages. So you can see here how the rolling circle replication is working. So one important thing is that in this process, every copy is made from the original template, original circle. There is no copy of a copy. So this is a really fundamental difference from PCR clusters. The main benefit of that is there are no clonal errors. In the PCR clusters, if the error happened in the first or second copy, that propagates, amplifies. Half of the spot has that, or whole spot has that error. And that's the sort of the biggest difference. You can have very accurate sequencing, but you still have errors in the array preparation. Also, because it's not PCR, there is no index scoping, so we have papers with 100 times less index misassignments with a single index compared to Illumina double index approach. I mentioned it's only 200 nanometer particles, so it's really, it can provide high density nanoarrays. That's how we reduce reagent consumption. These are small particles, but very bright, so we get a really focused, bright signal for easy imaging, low-cost sequencer, and a very low duplication rate. So all of this is our fundamental advantages that why our sequencer can provide the highest accuracy and the lowest cost. So using the technology over the years and investing in smart and efficient engineering, we were the first company that we've announced at the beginning of this year sub-$100 genome on our mainframe TX, T10, T20 sequencer. So that's a real achievement, from billions of dollars, and our first genome was $5,000 in 2010, when everybody else was 50,000. So we moved from 5,000 to soon will be 50. So it's crazy, crazy how much progress we made. And T7, it's sort of, as I mentioned, flexible. You can run one flow cell, and it's only $150. So I think T7 per genome, and this is all reagent cost. It's a real game changer. So is that the end of this technology? No, it's not. So we are doing, the arrays now that are, these 500 nanometers, but DNA nanoballs are 200. So we have a space, easy to put 4 more DNBs in between, so reduce cost reagent 4 times. So I really firmly believe, this is not just believe it, we're working toward, we'll get to $10 genome in a few years. So in terms of the genome sequencing, as I mentioned, Complete Genomics, we started with that goal to provide routine genome sequencing. We published this paper in 2010, $5,000 genome. And then, first familial sequence with Lee Hood, published that in Science, lung cancer, tumor normal. 50 genomes for the intellectual disability patients was a really good paper in 2014 that proves that whole genome is much better than exome. So, people didn't believe, but that was the paper that proved that. So when you take one of our genomes and run twice, even in this case it was different library, you have pretty good reproducibility. You should, as expected. At the beginning, it used to be that indels, we would have like 10% difference between 2 runs. We managed with PCR-free and other tricks to reduce that practically the same quality as SNPs. Only about 2% difference between 2 runs. I would like to see this to be 0. And the question is why there are these differences. They're not sequencing errors, that's for sure. So they are most likely somatic mutations that we pick in one run and not in the other. And we have solutions to sort of detect that. So how accurate are our genomes that are based on a completely PCR-free process when we compare them to the cluster-based methods? If we use GATK, so clusters are giving PCR clusters about 15,000, 10 to 50,000 indel errors. We are down to about 3,000. So there is a substantial difference in reduction, as expected, because PCR creates primarily indel errors. That's why people do PCR-free libraries. But when you do clusters to prepare arrays, it's not a PCR-free process. Only our process is a PCR-free process. So with machine learning, it's improved, but ours improved to indel sub-1,000 errors. It's a real accurate sequencing. The most accurate, I think, whole genome sequencing, because it's a real PCR-free. So we can see the benefit also on this medically relevant genes. We cover completely and without any errors 130 of them. Illumina is 127. So it's really good, but for any NGS, there are some genes, about 20, that we don't cover. PRN 150 is not enough to sequence some difficult genes in the families. So we need better genomes than today's best genomes, and we have a solution for that. So that solution we call Complete WGS, where complete genomics, everything has to be complete, so Complete WGS. It's easy to remember. Complete genomics, Complete WGS. So what is that? How do we do Complete WGS? We combine our real PCR-free WGS with the best accuracy with our single tube LFR WGS. This is a process that barcodes DNA, both are run on the same machine, both are PRN 150, but single tube LFR has a special barcode. So what this combination provides? It provides phased genomes, reduces errors, detects more structural variants, and covers most, if not all, of these blind zones, these 20 genes or so that we cannot sequence with the best PCR-free WGS. So we're automating or finishing that automation of this process on T7. So we'll be running 30 of this Complete WGS in 24 hours. T7 is so fast, PRN 150 in 24 hours. No other sequencer can do that. So, and what is the most important, it's affordable. Even though we do 2 libraries, it's only about $300 for the sequencing kits to get phased, accurate, higher quality genome. We really believe this will become a new standard. So how this single tube LFR works, how do we barcode? So, this was invented a long time ago, when it was 2005. But basically what we do, every read that comes from a long 100 kb, 200 kb fragment has the same barcode. And every other fragment, reads from that other fragment, get the same barcode different from other fragments. So we know we can cluster reads per each 100 kb molecule. So it's a really analogous to single molecule sequencing. We don't get continuous reads. We don't read, we don't exactly count triple repeats, but other than that, practically using same as single molecule sequencing. So this was published in Nature and in Genome Research, and all of this is available. So what's the advantage of single tube LFR? It's a single tube. It's like any other standard WGS prep. You can use same automation, same 96-well plate to make 96 samples. There is no drops. There is no oil. So what is happening, a long fragment just sort of wraps around the bead with the barcode. We have more barcoded beads in a tube than fragments, 3-4 times more. So that's why we get, most of the time, only one fragment per barcode. The 10x had something that they call link reads, but they would have 20, 30 fragments per one barcode. This is for the first time, single fragment per barcode. Most of the time, there is, this stochastic. And because of that, this is really sort of, for the first time, it's equivalent, or not equivalent, analogous to single molecule sequencing. It's a single molecule per barcode. So why is the haplotype phasing important? When we get multiple mutations per gene, especially if we consider a regulatory mutation, we really have to know, do we have one good protein and with good expression, or both alleles are, either bad protein or no expression. So this is really critical. And to get to the expression, the regulatory regions are mega bases downstream or upstream. So it has to be long context. You cannot do it with 10 kb phase context. So that's sort of why this every genome should be sequenced and be phased. It's so biologically important. So another advantage, I mentioned that most of these differences, if I run my genome twice, inherited variants should be the same. Why I get these differences? somatic mutations that we detect in one run and not in the other. So this method allows to differentiate inherited, so this is inherited het. You know, one haplotype has G, one has A, but when there is a somatic mutation, one of the haplotype has a second, you know, allele. So it's like a, you know, we see 3 haplotypes. So we can differentiate that using this technology and finally say, these are your inherited variants and these are your somatic variants. So in terms of the accuracy, this phasing is, you know, we phase almost every SNP. You know, the average fragment lengths are, you know, sort of in the order of 70 kilobases, and 50 for contigs, anywhere between, you know, 30 to 70 kilobases, megabases. So it's a very, very efficient phasing. So if you look on this chart, you know, one color block is one haplotype contig. You can see most of them, a whole chromosome arm is one phased haplotype. So it's probably the best phased genome, and it's affordable at the same time. So when we look, you know, some of you maybe, you know, heard Mike Snyder presented yesterday. We did his genome this way, and he was very, very excited about the accuracy and the phasing. So in this chart, we counted all the genes that have, you know, compound heterozygote, multiple hets. And, you know, 1,000 of them have them in both alleles. So there is a sort of risk there. Even if we focus on high impact variants, there is about 50 genes that both alleles are affected. So that's the benefit of phasing and a long haplotype phasing. So the phasing, the LFR provides another benefit. It helps cover difficult regions. As you can see here, everything is practically covered similarly, like you know, in general. But if you look individual genes, look at this SMN1. If we do pairing 150, us and everybody else, no coverage. We have good coverage with LFR, all of these examples. So because of that, we can now call a variant in a gene that before was not covered. That's another benefit. We can sequence and call variants in dark genes. So this is one of the other technology that DNBSEQ provides. It's our spatial solution that is providing subcellular resolution because these DNA nanoballs are 500 nanometers apart. We'll hear talk today about using this technology. So you use arrays for the spatial barcoding, but then you use our T7 for deep sequencing because you need lots of reads, so the cost-effective sequencing. So it's a really interesting how the same technology is used two different, at 2 different points to provide this sort of never seen before resolution in spatial that is subcellular. So we're happy to hear you know, use of that. So we need partners. With all the sort of technology that we have, we need ecosystem of people that develop different kits, different solutions, and we're happy to you know, work with them so that we can move the genomics forward, and you know, hopefully at the end, you know, prevent disease and get in, you know, longer, healthy lives. Thank you.